Hello, just thought I would go over a few review and practice questions about the blood and the heart for Unit 2, since the Unit 2 exam deadlines are coming up. And again, I will make these questions available on Blackboard. Remember, they're just some example ones for us to go through and kind of talk through how to approach some of these topics and questions. You're not going to have the exact same questions on the exam, but hopefully this will help you a little bit with understanding what you will see on the exams. And again, remember, in Blackboard, use those review activities that are available to you in the Chapter 17, Chapter 18 folders. Those have lots of practice items in there for you, both with the anatomy and with the physiology. And then don't forget to go back and use your Mastering A&P homework questions as well. I assign a lot of those because I think answering questions and making sure you understand why the right answers are the right answers, I think that's one of the best ways to study for an exam in any class, not just this one. Okay, so the first question we got here, practice question on blood functions. Which of the following is not a function of blood? And again, read questions carefully. Be sure you notice the word not in there. For many students, many students zip through exams much too quickly. You should not be taking one of these tests in 20 or 25 minutes. I mean, if you are, unless you are a speed reader and a genius, you're probably going through the test too quickly, not reading carefully. And you guys know you're in Biology 202, so you've already been through Biology 201. There's all kinds of new terminology in every unit in an A&P class. It's like learning a foreign language. You guys don't know this terminology well enough yet to zip through the tests. When you're learning new terminology, you have to read slowly and think about what you're reading to make sure you actually understand the question and all the possible answers. So okay, I'll stop my sermon now on that and go back to the question over here. Which of the following is not a function of blood? So which of these do we think? Protection, delivering nutrients to tissues. Yeah, protection, that is a major function of blood. Your white blood cells are in there to protect you against harmful invaders, germs. Delivering nutrients to tissues, absolutely, the blood does that. That's one of its key functions. They're highways that are delivering nutrients that have been absorbed from the foods that you've eaten to your tissues. They deliver oxygen gas to your tissues. Transporting hormones, yeah, hopefully you remember hormones travel through the blood to your tissues. How about C, excluding water from vessels to allow more room for cells? No, that's not going to be a key function of the blood, but those other things are. Formed elements. Remember, formed elements is another term for blood cells, and the reason formed elements is a preferred term is because a platelet is not a true cell, and really a red blood cell in some ways is not really a true cell either, because it doesn't have a nucleus. So sometimes all of these cells in the blood are referred to as formed elements. Which formed element is matched incorrectly to its function? All right, how about a lymphocyte, phagocytosis of viruses. Neutrophil, strongly phagocytic. Monocyte becomes a wandering macrophage in tissues. Eosinophil, important for combating parasitic worms, and also involves, that should be involved, not involves, in allergies. All right, if you've learned your functions, main functions of these cells, hopefully this is the one that jumped out at you. Lymphocytes don't do phagocytosis. One of the main functions of lymphocytes is they are involved in making very specific responses against very specific types of microorganisms that are invading the body. And some of your lymphocytes make antibodies. You may have heard of those before. Those are protein weapons that help you fight infections. But lymphocytes don't do phagocytosis. That's not one of their main jobs. All right, blood composition. Which of the following would not be found? There's that word not again. Would not be found in blood under normal circumstances. Key word there, normal. We have to read carefully. Water. Of course you have water in your blood. Now I will tell you, if I put a similar question like this on a test, I will have students who will say water is not part of your blood. Of course water is part of your blood. Water is part of your whole body. Water is the major component of the blood. Calcium ions. Oh yes, you have calcium ions in your blood. You guys have learned about how important calcium is. Insulin, do we have insulin in our blood? Yes. Insulin is a hormone that's important for glucose regulation. Microorganisms? No. Microorganisms are not in your blood under normal circumstances. Microorganisms in the blood, that's considered foreign, and they're eliminated by your immune system, we hope. Fibrinogen? Yes, remember fibrinogen is an inactive form of fibrin that's always present in your blood, and that whole blood coagulation pathway you've studied about converts fibrinogen to fibrin. Fibrin then forms a clot, that netting that generates the clot. But fibrinogen is always present in your blood, poised and ready to coagulate or clot your blood when needed. All right, hemoglobin. Select all that apply about hemoglobin. Okay, so remember with these select all that apply questions, you want to go down the list and just treat each one kind of like it's a true-false question. All right, hemoglobin, mostly composed of proteins. Yes, that's true. A hemoglobin molecule is primarily a set of 4 globin proteins that have joined together to form the hemoglobin. Contains 2 iron atoms per hemoglobin molecule. Yeah, that's actually false. If you go back and look, it's 4. Main role is transport of carbon dioxide. Keyword there, main. Now, hemoglobin does transport some carbon dioxide, but what is its main role is to transport what? Oxygen, that's right. Heme groups are degraded to bilirubin when our red blood cells croak, when they die. Yeah, that's where your bilirubin comes from. The heme part of hemoglobin, and that's not a protein, that's a different kind of organic chemical that's in there as part of the hemoglobin. But when that gets broken down, when your red blood cells die and they get gobbled up by macrophages and all their little parts get recycled, the hemes get converted to bilirubin when those red blood cells die. And then the liver takes up the bilirubin and sends it down into your intestinal tract for you to remove. That's the way the majority of it is removed from the body. A low protein diet could lead to deficient production of hemoglobin. Yes, hemoglobin is a protein. In order for you to make your own proteins, hemoglobin is primarily made of proteins. In order for your body to be able to make its own proteins, you've got to consume proteins. You need those amino acid building blocks from protein. So if you're on a low protein diet, you are at risk for not being able to make as much hemoglobin as you actually need, and that can lead to anemia. Hematopoiesis. All right, so a couple of H words in here. Hematopoiesis refers to what? Blood cell formation, which takes place where? In the bone marrow. And don't confuse that with hemostasis. Hemostasis is a term that refers to blood coagulation, keeping the blood vessels stable, the blood system stable. And hemostasis is not the same as homeostasis, that more general term that we've learned about keeping various functions and quantities of things within the body within a stable range. All right, so this question is about blood cell formation. Select all that apply about hematopoiesis. Let's go through these. A, does it occur in the liver? No, it occurs in the bone marrow. Hemocytoblasts are the precursors to all of your formed elements. Yes, these are a type of stem cell. So those are unspecialized cells. As they divide over and over and over again, they can specialize into the various types of formed elements. Most mature formed elements develop from lymphoid stem cells. That's false. If you go back and look at your diagram, you don't need to memorize the entire all of the pathways of hematopoiesis, but you should be familiar with which types of formed elements to send from your myeloid stem cells and which ones to send from your lymphoid stem cells. So if you go back and look, no, most come from the myeloid stem cells, not the lymphoid stem cells. Do platelets develop from megakaryocytes? Yes, they do. The megakaryocytes are these big giant cells, and they break apart to form your platelets. Erythrocytes and neutrophils have a common ancestor beyond the hemocytoblasts. So that one might be a little bit tricky, but if you go back and look, hemocytoblasts, they divide. Some of those become your lymphoid stem cells, some become your myeloid stem cells. As myeloid stem cells continue to divide and specialize, some of those will become erythrocytes, and yes, some of those are going to become neutrophils. So even though they have very different functions, they're coming from the same myeloid stem cell ancestor. So that one is actually true. So our correct answers B, D, and E. Select all that apply about a patient who is experiencing difficulty breathing due to years of smoking. All right. Difficulty breathing due to years of smoking. This is one of those kind of critical thinking, clinical application questions. All right, cells in the heart are going to detect low oxygen levels and secrete EPO, erythropoietin, in response. So this is where you have to be careful, because this part is to detect low oxygen levels and secrete EPO in response. That does happen when you have low oxygen levels, like if you've been smoking for most of your life and you're not able to take oxygen into the body very well. But cells in the heart don't do that. Remember, cells in the kidneys are responsible for that. EPO targets immature forms of erythrocytes in the red bone marrow. So is that true or false? Yeah, that is actually true. EPO, its job is it stimulates more of these immature erythrocytes to go ahead and specialize into full-blown functional erythrocytes or red blood cells. And then that's going to help you. More red blood cells will be able to bind more oxygen in your poorly functioning lungs. All right, this patient may develop secondary polycythemia. Yes, that is true. Polycythemia, excess red blood cells. And if that gets triggered due to low oxygen levels, like let's say you're living at a high elevation, or if you're not taking in oxygen well due to poor lung function, then you can develop what's called secondary polycythemia. EPO production will decrease in this patient. No, we've just been talking about how it's going to go up. Increased EPO will stimulate an increase in all formed elements of the blood. No, I mean, you can tell that by the name. Erythropoiesis, erythropoietin stimulates erythropoiesis, and that specifically refers to red blood cell formation and not formation of your other formed elements. The patient's blood may become more viscous. That's going to be true. If you have secondary polycythemia, you have more red blood cells in the blood, it can become thicker. So B, C, and F on that one. Okay, what are each of the following that may cause anemia? Poor iron, and again, this is multiple response. Poor iron absorption. Yes, you need iron, absolutely. Iron is part of your hemoglobin. Iron is required for you to be able to bind oxygen on your hemoglobin and carry it throughout the body. So if you don't have enough iron, you can become anemic pretty quickly. Internal hemorrhage. If you had ruptured blood vessels, you were bleeding internally, absolutely, you could become anemic due to that. Acute lymphocytic leukemia. Yes, now what is acute lymphocytic leukemia is where you have an overgrowth of immature lymphocytes. This is coming from your lymphoid stem cells or some sort of immature form of lymphocytes that are overgrowing. They've become cancerous, and when you have that going on, that tends to reduce the quantity of other formed elements, like your bone marrow becomes concerned with primarily making these abnormal white blood cells, and your other types of formed elements are not going to be made properly, including your red blood cells, which can make you anemic. Are you going to have increased EPO? Would increased EPO production cause anemia? And hopefully you're thinking no. EPO stimulates more red blood cells to be made, not fewer. A lack of intrinsic factor. Yes, if you go back and look, intrinsic factor is necessary for proper absorption of vitamin B12, and if you don't have adequate vitamin B12, you're not going to make red blood cells properly. All right, vitamin B12, you need that for DNA synthesis. So if you can't do your DNA synthesis properly, you're not going to be able to divide your cells properly. All new cells need DNA. And so if your cells aren't going to divide properly, you're going to become anemic because you're constantly having to crank out more and more and more new red blood cells to replace the ones that are dying. What if you had an infection of your red bone marrow? Yeah, infections of the bone marrow can cause you to produce fewer formed elements, including your red blood cells. Okay, so lots of those could cause anemia. What are some other things like exposure to toxins and pesticides? A low protein diet. If you're not getting adequate protein, you can't make hemoglobin properly. So go back and review the different things that can cause anemia. Put these steps of hemostasis in order, an ordering question. All right, I'm going to have to get out my pen for this one. Hemostasis, again, that's another term for blood clotting, blood coagulation. Hopefully I can get my pen to write on my iPad here. It doesn't always work real well. Okay, so what are we looking at here? What do we think's going to happen first if we read through there? All right, hopefully you're looking at C here. A blood vessel gets torn. That's going to be the thing that's going to get this party started here. So we're going to say that's number one. All right, so when a blood vessel is torn, what's the next thing that's going to happen? It gets torn. Blood vessel starts to spasm. Remember, that's almost instantaneous. That's gonna, if you have a tear or a crack in a blood vessel, the spasms, the smooth muscle in the blood vessel starts contracting to try to close off the tear. Let's see what's gonna happen next. I was looking for platelet plug formation, but that's not actually part of this question, it's part of the answer, so we'll skip over that one. Then we were going to say the coagulation pathway gets triggered. And if you go back through your intrinsic pathway, your extrinsic pathway, remember, you've got these two different pathways, and the goal is to make this prothrombin activator. That's where both of those pathways are leading, so that's going to be number 4 on our list. Prothrombin activator, what is that? That's an enzyme, and it converts prothrombin, which is always in your blood, that's in an inactive form. Prothrombin activator converts prothrombin to thrombin. That's a key, crucial step, so that's going to be step 5. Thrombin then converts fibrinogen to fibrin. So you've got this multi-stepped pathway that amplifies to allow you to make a blood clot. You may be wondering, why in the world is this so complicated? Well, blood clotting is not something you want just happening randomly or you know, at the least little minor thing. It needs to be very carefully controlled so that you only generate blood clots when needed. And of course, in this unit, you guys have learned about some of the consequences of when you have clots that form when they're not supposed to. All right, so the last thing, once fibrinogen has been converted over to fibrin, those fibrin threads are cross-linked together and the clot contracts. So that's going to be step number 7 there. So this is one of those pathway things. You just kind of need to know the sequence of events for how that happens. All right, which of the following digests the fibrin threads of a clot? So when you have a clot that's formed, you don't want that clot to be there permanently. Once your blood vessel has been repaired and so forth, we need to dissolve the clot. And so what actually breaks the clot down? So there's something, one of these three things is an enzyme that digests the clot. And hopefully you're thinking B, plasmin. These other two things are involved. Remember, tissue plasminogen activator is what converts plasminogen to plasmin. Whenever you see this ogen on the end of things, that's generally the inactive form, and then it gets converted over to the active form. And then the plasmin then will break down the clot when we're ready to do so. The patient travels on a long flight to Asia and starts to experience leg pain. 3 days later, the pain stops suddenly, but then the patient has extreme difficulty breathing and dies. What has likely happened? This is not uncommon for people who take very long flights across the ocean. And hopefully you are picking pulmonary embolism here. So a clot developed in a vein in the leg. When you're sitting on a flight for a long period of time, the blood flow is slow through veins anyway, and slower blood flow or blood that is flowing more slowly is more likely to clot. If that's a thrombus, a clot is a thrombus. If that breaks away, it becomes an embolus. And that embolus can travel to the heart. The heart's going to pump it over into the pulmonary circuit. And if that embolus gets lodged in one of the veins of the lungs, it can cut off the blood supply to one of the two lungs, causes problems with blood flow in general and pressure problems and so forth, and can lead to death. That's a pulmonary embolism. What's a myocardial infarction? That's where you have a thrombus or a blood clot in one of the arteries supplying the wall of the heart itself. That's another name for a heart attack. Stroke, that's where you have a thrombus that cuts off arterial blood flow to some part of the brain. Angina pectoralis is just referring to pain in the chest, and that's like when you have like a partial blockage of an artery in the heart. That is, you're not having a full-blown heart attack, but you're experiencing pain because you have part of the wall of the heart is not getting as much oxygen as it needs, so you're experiencing pain from that. Okay, impaired liver function can lead to problems with coagulation. True or false? Hopefully you're saying true on that. Why is that true? Because the liver makes most of your coagulation factors. When the intrinsic and extrinsic pathways, all those blood clotting proteins that are part of those pathways, most of those are made by the liver. Blood type. Gomer has type AB negative blood. Which of the following is true? He has AB negative blood. Is he the universal donor? No, he's not. He's not the universal donor. If you have type AB blood, that means you have A antigens and B antigens on the surfaces of your red blood cells. So you can't donate to everybody else because any other blood type is going to react to one or both of those antigens. Does he have RHD antigens on the surfaces of his red blood cells? No. If you have those on the surfaces of your red blood cells, then you would have positive type blood. He has both A and B antigens on the surfaces of his red blood cells. That's true. His blood contains anti-A and anti-B antibodies. Remember, the antibodies are the weapons generated by your immune system to things that are foreign to you. So if you have type AB blood, you should not have anti-A and anti-B antibodies, because those antibodies would attach to your own red blood cells, and that would not be a good thing. So our answer there was C. A pregnant woman would be given RhoGAM. When are those RhoGAM shots given? She is Rh negative and possibly carrying an Rh positive fetus. That is correct, and you guys can read through the other three answers there, but that's when RhoGAM is given, and that is primarily given to protect the next fetus she might have if she gets pregnant again and that fetus is Rh positive. If she wasn't given RhoGAM during the first pregnancy, she may have generated antibodies of her own against the Rh positive fetus's blood cells. Those antibodies can go into the fetus the next time and wind up harming or killing the fetus. So RhoGAM is a protective type of medication that's given to prevent that. Okay, getting into blood flow, pulmonary and systemic circuit. Select all locations where you would find deoxygenated blood. So this is a pretty good little critical thinking question here. Let's go down the list. Again, treat these like true-false questions. Superior vena cava, will that have deoxygenated or oxygenated blood? I'm going to turn my pen on here so we can check these off as we go through them. Hopefully you're thinking yes on that one. Superior vena cava is bringing, it's a vein bringing deoxygenated blood back to the heart, from the head, the arms, the shoulders. The left atrium over on the left side of the heart. No, you have oxygenated blood on that side of the heart. Remember, the left atrium receives blood coming back from the lungs after it's been oxygenated. Pulmonary trunk, yes, that blood is deoxygenated. Blood has flown from the right atrium to the right ventricle. The right ventricle pumps that blood up into the pulmonary trunk, which is going to then send it off to the lungs. Pulmonary arterioles. The pulmonary arterioles. Yes, remember the pulmonary circuit is opposite of the systemic circuit. Arteries and arterioles in the pulmonary circuit have deoxygenated blood. Veins and venules have oxygenated blood in your pulmonary circuit. So that's going to be a no. I mean, I'm sorry, that's a yes. And your pulmonary venules are a no, because pulmonary venules have oxygenated blood that's on its way back to the heart, so the heart can pump it out to the rest of the body. How about the ascending aorta? No, the aorta is going to have oxygenated blood. If it doesn't, you're in big time trouble. Coronary sinus, you guys remember where that is? That's kind of a big, thick vein on the posterior side of the heart, and it is bringing deoxygenated blood that has drained off of the walls of the heart itself and returning it over to the right atrium. So that will have deoxygenated blood. How about the circumflex artery? No, the circumflex artery is one of the coronary arteries that's supplying the wall of the heart itself, and so it better have oxygenated blood in it. Otherwise, the myocardium of the heart is not going to be too happy about that. A, C, D, and G. Okay. Select all locations where you would find oxygenated blood. So the opposite question here. And yeah, when you guys are studying, you want to be sure you understand the pulmonary circuit versus the systemic circuit, where you have oxygenated blood versus deoxygenated blood. You want to review that whole flow of blood through the entire heart, from chamber to chamber, what valves are involved, the major blood vessels. You want to make sure you understand how all of that works. You got to kind of you can't just memorize words and definitions to make it through this unit. You got to actually study the step-by-step processes. All right, all locations where you would find oxygenated blood. Aortic arch, absolutely. That's a gigantic systemic artery. It's going to have oxygenated blood. Left ventricle, yes. The left ventricle is going to pump oxygenated blood out of the heart and towards all of your tissues, other than the lungs, other than the pulmonary circuit. Right marginal artery, yes, that's one of your coronary arteries that's bringing oxygenated blood to the myocardium, the wall of the heart itself. Left pulmonary artery, that's going to be a no. Remember, your arteries in your pulmonary circuit have deoxygenated blood. Arteries carry blood away from the heart. Your pulmonary arteries are carrying the deoxygenated blood away from the heart toward the lungs so that it can be reoxygenated. Arterioles of the spleen. Yes, any artery, arteriole in the systemic circuit is going to have oxygenated blood. The hepatic portal vein, no, that's actually, you guys, we haven't really studied the hepatic portal vein yet, but that's hepatic means liver. Vein refers to veins that are part of the systemic circuit have deoxygenated blood, so that's going to be a no. I wouldn't ask you that on this test, though. Celiac trunk, this is a question that was used when we did all of cardiovascular as one unit, so don't worry about celiac trunk yet. It's coming up soon, though. That's actually an artery that you'll be learning about in Unit 3, so it does have oxygenated blood in it. Internal jugular vein, that's you guys know where your jugular veins are, and those are going to have deoxygenated blood in them. And again, with that particular question, that question was written back when we used to do all of cardiovascular together as one unit. So as far as what you guys need to know for this test, deoxygenated and oxygenated, just think about the pulmonary circuit and the heart chambers, the coronary blood vessels around the heart, and then those major blood vessels that are branching off of the heart, your inferior and superior vena cava, pulmonary trunk, the parts of the aorta, and so forth. All right, heart coverings and layers. Put these in order from superficial to deep. Okay, well, these ordering questions again. If you're thinking about the coverings around the heart, which of those would be the most superficial? Thinking about the different layers. Hopefully you're thinking fibrous pericardium. That's that outermost covering. All right, and then the fibrous pericardium, that's that tough, dense connective tissue covering around the heart, very tough. And then sitting along the inside of that, kind of lining the inside, what are we going to have? We're going to have our parietal pericardium. All right, and then if we keep going, what are we going to hit next? Visceral pericardium. That's remember, the visceral pericardium is actually in contact with the surface of the heart itself. Keep going deeper. Now we're in the wall of the heart itself. Next layer is going to be the myocardium. That's the muscle layer. Endocardium, that's the innermost layer of the heart that's lining the chambers of the heart. And then finally you would hit blood inside a chamber, so we'll make that number 6. All right, so be sure you've got a good visual from superficial to deep of these different layers and coverings. All right, blood leaking out of the aorta and into the pericardial fluid may cause this condition. Which of those? And hopefully you're saying cardiac tamponade, which that's an accumulation of fluid within the where you get blood or excess fluid in between the visceral pericardium and the parietal pericardium, and that puts lots of pressure on the heart. It makes it difficult for the heart to contract properly and circulate blood. Okay, what are the connective tissue strings that pull on the AV valve cusps, your atrioventricular valves, when the ventricles contract? And what are your 2 AV valves? Hopefully you're thinking bicuspid and tricuspid. All right, and when the ventricles contract, what are the strings called? Hopefully you're thinking chordae tendineae. Answer D. Remember the papillary these are connected to the papillary muscles. The papillary muscles contract, they pull on the chordae tendineae, and those chordae tendineae are attached to the cusps of the valves, and they pull those cusps shut. So when the ventricles contract, you close off the opening between the atrium and the ventricle. All right, the cusps of this valve catch blood flowing downward in the pulmonary trunk as the right ventricle relaxes. If your right ventricle is relaxing, what valve is going to make sure you don't have blood backflowing from the pulmonary trunk into the right ventricle? Now, hopefully you're thinking B, the pulmonic SLV, semilunar valve. Remember, your semilunar valves are shaped like little cups, and the cusps, of the valve are shaped like little cups, and that helps catch blood that just by gravity is trying to backflow toward the right ventricle or the left ventricle over on the aorta side of things. And so they're shaped like that to help prevent the backflow of the blood. All right, cardiac muscle fibers receive their oxygen gas and nutrients by diffusion out of the left ventricle and into the myocardium. Where do your cardiac muscle fibers within the wall of the heart itself in the myocardium, where are they getting their oxygen and nutrients? Is it just diffusing out of the left ventricle? Left ventricle's got lots of oxygenated blood in it. Now, hopefully you're answering false on that, because the whole purpose of your coronary circuit, all these coronary blood vessels, coronary arteries, and veins, is to make sure you're circulating blood to the wall of the heart itself, the myocardium. The myocardium cannot receive all of its oxygen and nutrients just by simple diffusion out of the left ventricle. If you just think about that, that would be extremely difficult. So you've got to actually have arteries and arterioles and capillaries delivering the blood to the wall of the heart itself. And your myocardium is very metabolically active. It's a very active, hopefully it is. Otherwise, you know, when your heart stops beating, that's when you die. So uses lots of energy. So the wall of the heart itself needs lots of oxygen gas, needs lots of nutrients to make the ATP it needs so that your heart wall can contract. All right, a question about the cardiac muscle fibers or cells themselves. Select each of the following that is true about cardiac muscle fibers. So go back and review some of those basic characteristics that we learned about them. Are they branched? Do they have branches? Yes, they do. So there are branches connecting one fiber to another. Do they have gap junctions that allow fluids and action potentials to flow between the cells? Yeah, those are like little openings between individual cardiac muscle fibers. And yes, your cardiac muscle fibers have those. Your skeletal muscle fibers you learned about in Biology 201 do not. And remember, in order for your heart to beat as a unit, you need these action potentials that trigger contraction to spread very rapidly across the entire wall of the heart. And so the gap junctions help that happen. Are your cardiac muscle fibers controlled by motor neurons? No, they are not controlled. Motor neurons control your skeletal muscles. The heart rate and the force of contraction of the heart are controlled by your autonomic nervous system. Okay, they're not controlled by the motor neurons that stimulate contractions in skeletal muscles. Do they require large amounts of oxygen gas? Absolutely. Yes, your cardiac muscle fibers consume tons of oxygen gas because they've got to make lots of ATP to power all this contraction they have to do. Do they contract together as a large unit? Yes. Are they controlled voluntarily? No. Hopefully you're not having to rely on voluntary control of your cardiac muscle fibers. That would be kind of difficult. All right, our intrinsic conduction system. So go back and review all of the parts of the intrinsic conduction system. That's the electrical system of the heart that starts the action potentials that will spread across the entire structure of the heart. That's another term for like the electrical wiring of the heart. And the electrical signaling, the action potential that passes through this conduction system, has a specific pathway that it normally follows if you're nice and healthy. And so you want to know that sequence. So where does the electrical signal or action potential start? Hopefully you're picking, I get my pen to come up here. Hopefully you're choosing SA node for that one. number 1 there, and then the signal passes over to the AV node. That's number 2. The AV node is up there in the right atrium, pretty close to where you have that opening that leads to the right ventricle. And then it's going to spread down through the AV bundle. The AV bundle divides up into the bundle branches, and those are located in the interventricular septum that's located between the little wall between the left and the right ventricle. And then finally, from the bundle branches, it spreads out into the Purkinje fibers that carry it up into the walls of the ventricles. So that would be our correct order. So again, go back and study the pictures, the diagrams, make sure you can follow that sequence of events. You want to go back, you want to study the entire cardiac conduction, the whole, one whole round of the cardiac cycle. You want to know that whole thing step by step. You know, you should be able to diagram it out. You should be able to write it out step by step what's going on. Okay, intrinsic conduction system. If the normal pacemaker of the heart fails, then this structure takes over. What's your normal pacemaker of the heart? And again, hopefully you're thinking SA node on that one. The SA node automatically undergoes an action potential, starts a new round of electrical signaling periodically. And so it's the pacemaker of the heart. If your SA node stops functioning properly, it doesn't mean your heart's no longer going to automatically be able to contract. The AV node is what takes over if you're a normal, healthy person. All right, during the cardiac cycle, which of the following happens first? Which of the following happens first? All right, so we got a couple of things on there about the ECG. So let me draw out like one round of a ECG cycling. P wave. Oh, come on. Pen, let's go. My pen doesn't like me today. There we go. Okay, here we go, finally. P wave kind of looks like that, and then you got a QRS wave. This doesn't look so good, but there's your QRS, and then over here you got your T wave. So that's what you would see. Remember that an ECG, an ECG is measuring the electrical activity around the heart. It's detecting the action potentials. It is not detecting the contraction. That's muscle activity. That's the actual the muscle fibers themselves shortening and causing a heart contraction. Okay, so the ECG is just detecting the electrical signaling that causes the contraction. So which of these things is going to happen first? A P wave on an ECG, contraction of the atrial myocardium, a T wave on an ECG, contraction of the ventricles. All right, so let's think about our story over here. When you see a P wave on an ECG, what does that represent? That P wave on an ECG just lets us know the walls of the atria, right and left atrium, are depolarizing. You've got that electrical activity that's spreading across the atria. Okay. All right. And what does that trigger? The electrical activity that you see with a P wave causes the atrial myocardium to contract. Your right and left atria contract. So the P wave causes atrial myocardium to contract. And then what does the QRS wave represent? That's representing the depolarization of the ventricle. So that electrical activity, that action potential, then spreads across the ventricles. And what does that trigger? Once that happens, then the ventricles contract. Okay, and then what's the T wave representing? The T wave is representing the repolarization, the walls of the ventricles going back to normal. So which of the following on our cardiac cycle happens first? hopefully you're thinking A, P wave on the ECG. So remember, the SA node starts the whole depolarization action potential process. And then that leads to a P wave if you're detecting this with an ECG, and that triggers the atria to contract. All right, which of the following happens after the QRS wave on an ECG? Select all that apply. Happens after, and we're just thinking about one round of the cardiac cycle. So would we pick A, depolarization of the SA node? No, that's the very first thing that happens in the cardiac cycle. So no, we're not going to pick that one. All right, closure of the AV valve. So you got to really know your cardiac cycle here to answer this one. QRS wave causes, that happens, and then the ventricles contract. Remember, when the ventricles contract, those little papillary muscles contract, pull on the chordae tendineae, and the AV valves close. So yes, B will happen after the QRS wave. Depolarization of the atria, no, that happens after the P wave. Actually, when you have a QRS wave on an ECG, yes, you're seeing depolarization of the ventricles, but the atria are also repolarizing, but they're doing that at the same time that the ventricles are depolarizing, so they show up together on the QRS wave. All right, so conduction of an action potential down the AV bundle. All right, your QRS wave indicates depolarization of your right and your left ventricle. Well, all right, for your QRS wave to occur, that action potential has already been down the AV bundle. It's been down the bundle branches up into the Purkinje fibers, and then it has spread across the walls of the ventricles. So no, this would not happen after the QRS wave. This would happen before it. Ventricular systole, what does that stand for? Ventricular contraction. And yes, that's going to happen after the QRS wave. That electrical activity is stimulating that. Opening of the semilunar valves. Yes, remember, it's the pressure of the ventricles contracting and squeezing the blood is what pushes your semilunar valves open, the pulmonic and the aortic semilunar valves. All right, so B, E, and F on that one. Okay, the lub heart sound occurs. Okay, answer popped up. The answer to that one is C, as the ventricles contract and the AV valves close. When you hear that, your heart sound is lub-dub, and the lub part is when the ventricles have contracted and caused those AV valves to close. All right, when ventricular pressure exceeds aortic pressure, what's going to happen? So your ventricles are contracting, pressure's going up, they're squeezing the blood. And think about your aorta up here above the heart. The aorta has a certain amount of blood pressure within the walls of the aorta. And you've got that valve in between. You've got your aortic semilunar valve. So what's going to happen to get that blood to flow from the left ventricle and actually up into the aorta? How are you going to get that valve to open up? Or you're going to, in order for that to happen, pressure in the left ventricle's got to be higher than it is up in the aorta. And so once that happens, the aortic semilunar valve is going to open. So hopefully you were thinking D on that one. All right, so those were just some example questions, and hopefully I've kind of, there at the end, hopefully I'm kind of enforcing with you, you need to be able to think through a single round of the cardiac conduction cycle and what's going on with that. Those are just some example questions. Be sure you use the review activities in your Mastering A&P homework. Return to those for other good ways to practice for the Unit 2 exams. Good luck. If you have any questions, again, I prefer that you post things on the discussion board so that everybody can benefit from your question and the response. You can also, of course, message me as well. So good luck with the exams.